Hello everyone, welcome to today's live broadcast, Genomes for Medicine. My name is Harpinder from Illumina and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. We have a few important announcements before we begin. The webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking the green Q&A button on the lower left of the presentation window. We'll try and answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right hand of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button on the top right of your presentation window or submit your problems through the green Q&A button on your left. This is an educational webinar and this offers free continuing education credits. After the webinar is over, please click on the CE button located on the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce our today's speaker, David Bentley. David is the Vice President and Chief Scientist at Illumina. He has played a leading role in the Human Genome Project and related international consortia to characterize the human sequence variation, including the SNP Consortium, the HapMap Project, and the HapMap Project. His long-term interest is the study of human sequence variation and its impact on human health and disease. His current research focuses on fast, accurate sequencing of human genomes and adoption and benefit in healthcare, with early applications including rare genetic disease and cancer. These applications are exemplified in the goals of the 100,000 Genomes Project, a partnership that includes Illumina, Genomics England, and the NHS England. With that, a very warm welcome to David, and to everyone listening. I hope you will enjoy the presentation. Over to you, David.
Hey, thank you very much, Harpinder, for that kind introduction. I'd like to add my welcome to everybody who is present for this seminar today, and I'll get right to it. I'm particularly keen to share with you some of the insights that we have gained as a result of collaborations which have really been geared towards improving human health by unlocking the power of the genome. It really stems from the Human Genome Project, the sequence released from that, and how we can put it to good use, ultimately in medical applications as well as research. I'll deal with four parts. I will briefly go through some concepts which underlie this work. I will describe something of the recent technology developments and how we monitor what's going on, touch on working at scale, which is particularly linked to the 100,000 Genome Project and other projects around the world of similar scale, and finally just draw a few conclusions to stimulate further questions and thoughts. So let me start with concepts. For some time, I think this vision of precision medicine has been evolving, and I think it's exemplified by this slide. It's a vision shared by many organizations. And it starts, if you will, at the bottom of the slide by the patient. This is the timeline of an individual as they go through life, from birth, indeed from conception. And you can see through the timeline a series of events that take place throughout this person's life. Unfortunately, this person gets cancer at one point during their life, and you can see a series of events which take place in the course of managing or relapsing, and ultimately the death of the individual as a result of the cancer. On the bottom of the line, you see a number of different arrows of different colors, and these indicate points at which we can make measurements of the individual, clinical observations or molecular measurements or sequence information, which can be collected at various points throughout a person's life. And they serve as important metrics to correlate with the clinical observations and the disease, so that we can understand better what the readout of a patient from their blood samples, from their DNA, or for whatever it may be, how they correlate with our understanding and our ability to either assess, diagnose, or indeed provide a prognosis for patients. And by exploring in detail a patient such as this, we can then extrapolate that information for benefit to future patients like them. As you can see, the patient information is optimally accumulated in a knowledge base, possibly in a cloud or a private cloud. And that information should then be available both to support research on the left. Individuals can access the data to support new findings, new discoveries, and on the right, by clinicians directly perhaps to help them in their treatment choice or other aspects of managing the patient's condition. And this whole ecosystem is one which is a long-term program, a collaborative venture with different countries and different organizations. But the vision is essentially the same, and it's a very shared vision across the world. Let me start with the concept which has really introduced the use of whole genome sequencing for genetic diagnosis. This is not entirely about whole genome sequencing, but my talk will focus predominantly on whole genome sequencing and mention other sequencing methods as appropriate. We have an undiagnosed condition. A male child presents with developmental regression to two unaffected parents. And essentially the concept is that by sequencing the genome of this individual, in this case in a 4-day turnaround time, which is a clinically relevant turnaround time, we can gain the complete information from that individual's genome, which is potentially responsible for what looks like a genetic condition. This is how we then filter through the whole genome from looking at the 3 billion bases or 3 million variants or so. We can examine transcripts which have particular variants which may be of interest and gradually filter down by asking additional generic questions of the genome. For example, we're expecting 2 variants. We're expecting a very low frequency mutation in the population. We can predict function, and we can examine whether a gene is previously known to be involved in a disease. And as you can see there, as you go through these various questions, you very quickly filter down. In almost real time, a few clicks of the mouse will actually lead you to a few, and sometimes one, a really strong candidate, which is a mutation which is in a relevant gene with a relevant function. And the important thing is to correlate that variant with the condition to confirm the diagnosis. And this particular illustration here is of a novel variant which correlated both with the genetics and the apparent function of that gene, which led to a diagnosis. A collaboration with Stephen Kingsmore, who is now at the Rady Genome Center. So this illustrated the application of whole genome sequencing. The model applies very well for whole exome sequencing as well, from doing a genome-wide search to find a point mutation which causes disease and offer a diagnosis, which is a great benefit to the family. To look at how this applies more broadly, I show you here a table of several studies which have really embraced that same method, that same approach. And you can see here that we've moved from single anecdotes which were reported as one-offs or N of 1 cases, through to looking at small or middle-sized cohorts now to examine how often does this paradigm actually hold. And you can see here that from a single test, from a whole genome or whole exome, they're lumped together in this set. We're looking at not 1 or 2, but we're looking at 25 and 30, and in some cases over 50% of the cases of previously undiagnosed conditions are actually amenable to interpretation and diagnosis by this method. And this illustrates the potentially important, powerful nature of the universal test, looking at the whole genome and seeing how we can apply it to these clinical diagnoses of different clinical types. Moving on to cancer for a moment, as I mentioned, this is another early interest. A similar anecdote reported here illustrates that in this case, a recurrent glioblastoma, which was not responding to treatments, was actually biopsied and sampled. In this case, 3 biopsies were sampled. Again, in a clinically relevant turnaround time. This is actually 10-day turnaround to get the 3 results. And one of the interesting things about this case is not only the clinical turnaround time was, again, relevant to the management of the condition. We returned the data back before the patient was ready for treatment. But also, in particular, in this case, we found not so much single nucleotide changes, and I'm talking here about somatic changes in the tumor DNA as compared to the normal DNA. We saw in this case copy number variants throughout the genome as well as other variants. And you can see here in particular, one of those copy number variants really amplified a particularly important gene involved in growth control. And in fact, several CNVs actually amplified several genes involved in the same signaling pathway. And this is illustrated here. One of the important points here, of course, is that as you find mutations in the genome without a prior hypothesis, you suddenly find not one but several hits on a particular biologically important function. But you also find that you are essentially suddenly harnessing years and years of biological research that has gone into understanding the signaling pathway, and immediately that information is accessed and made available to benefit the interpretation of this patient. And you can see here these 3 genes which were amplified in the genome, among other mutations, all lead to stimulating and overstimulating the same signaling pathway, which leads to cancer. Moreover, a very important finding from this pathway is actually in looking up the available drugs. You can actually see there that not 1, but actually there were 3 FDA-approved drugs already available, all of which would act to block this pathway, and none of which had been used previously on this particular case. So this really embraces the paradigm that sequencing of a tumor can actually potentially inform therapy. And this echoes the original time this paradigm was put forward by Mark O'Mara back in 2009. This particular collaboration is with Charlie Swanton in the CRUK here in London. Looking on to other aspects of whole genome sequencing, cancer is a complex condition. I want to show you a few examples of other collaborations where we have explored different readouts from the same whole genome sequence data, which reflects on our description or our characterization of a condition. This example here is a collaboration with Newcastle, where we use whole genome sequencing of acute lymphocytic leukemia, ALL samples. And in this case, we show the high hyperdiploidy, which is a characteristic of some childhood ALL, and is indeed an indicator of good progress. A simple readout, an alternative to cytogenetic testing. In this example, we use whole genome sequencing at multiple time points in the course of a chronic lymphocytic leukemia, and this reveals two things. You can see in the first two time points, essentially different subclonal populations can be identified from the whole genome somatic mutation profile, and the red subpopulation dominates by the time this next treatment is given, and is largely removed by a successful response to the treatment, as you see in the third point. When you look ahead to fourth and fifth time points, some few months later in the individual's life, you can see a remission, which is characterized by a re-expansion of subclonal populations, but in this case, a different subclonal population characterized by a different profile of somatic mutations has now arisen. Ironically, however, at the time when we sequenced the patient when it was deemed to be in remission, you could actually see from the sequence data that we carried out, the whole genome sequence, that already the new profile of subclonal populations was expanding just at the time when the patient was declared well. And clearly in this case then, if you looked at the molecular signature, you would not conclude that this patient was exactly in remission. Clinical remission may be, the symptoms were alleviated, but the disease is already present at the molecular level, and this would have been an opportunity to intervene earlier with the benefit of molecular data. So this just illustrates then a time course of a cancer patient where we can use sequence information, the somatic mutation profiles, both to alter our understanding of the remission and look in general at the response of the individual to particular treatments. I'll come back to that concept in a moment, but I wanted to move on just for a second to a couple of signatures. Again, the opportunity to look at the entire genome and all the mutations in the cancer at a particular time point sometimes reveals particular context. This particular example shown here is an example of two contexts from two patients, where a majority of C to T changes in pyrimidine dimers typically is a signal of the exposure to UV light. And this is essentially, of course, a precursor to skin cancer, or a mechanism by which skin cancer arises. On the top, you see a signature which is different if you pooled all the mutations in a case which has been exposed to cigarette smoke. And these signatures are both essentially embedded in the somatic mutation profiles, which can be extracted and characterized quite quantitatively. Once you have a whole genome or a genome-wide data set of all these mutations, you really can look at the signatures of the exposure to the environmental factors which gave rise to the cancer, which may be useful information. Or you can also see signatures of defective DNA repair and other mechanisms which, again, may be relevant in terms of at least assessing the prognosis for the cancer. It's important to understand these is a research finding at this stage, but the availability of such information and making it readily available, I will return to at the end of this presentation, but making it readily available is important if we are to understand better how to use such genomic information in clinical reports and interpreting an individual's molecular profile for the benefit to the patient. Another example of such signatures, both the last one and the present one, in this case come from Mike Stratton's group at the Sanger Institute, just nearby. This is an example where, looking across an entire genome, you see all the mutations, somatic mutations, plotted across the entire genome. But you see one area of the genome where there really has been a catastrophic event, contagious. These are called rainfall plots, and this is a rainfall or a cloudburst, if you will, of a part of a chromosome which has really been hit by one particular mutation type. And this is in a BRCA1 mutated tumor, breast cancer. And clearly this information is not necessarily available from standard tests and traditional tests. It's a new kind of readout, but we can learn whether we can find this information useful in amending our diagnosis. Consider if you'd done a somatic panel, you'd seen a few mutations, and you'd returned a verdict on this breast cancer, but you'd completely missed this catastrophic event unless you have a genome-wide test, such as a whole genome, or in some cases a whole exome. Another important finding, just to reflect on this for a moment, is, of course, as you move outside the genes, it's the non-coding mutations also play a very important role in the derivation of cancer. This is a well-known example where whole genome sequencing, as opposed to exome sequencing, reveals formation of a novel transcription factor in the TERT gene, and actually leads to, in this case, a mutation which, when you survey other examples of melanoma, you actually find not only is this mutation present in other melanomas, but it's present actually at the same frequency as the very well-known BRAF V600E mutation. So these non-coding mutations are not edge cases. They are essentially going to contribute very significantly to our increased ability to understand and interpret individual cancer cases, and indeed potentially to classify them as a prerequisite to possibly improving the management of the condition. One more paradigm I'll just evolve for you is the diagnostic yield, which I showed for the rare genetic disease, that we're moving from anecdotes to a quantitative number of cases where actually you can characterize them. Here is an example, an early example from the ALL study, where 22% of all ALL cases are unexplained at the molecular level at the beginning of this study. They're either completely normal or some unexplained apparent rearrangement by cytogenetic testing. And this collaboration, again with Newcastle, we actually took the entire fraction of the unexplained cases at the molecular level and actually found an insightful mutation. Multiple new genes were identified in every one of them by whole genome sequencing. And so effectively the diagnostic yield, or the detection of clinically relevant mutations, some of which are still undergoing further validation, of course, and functional proof, but nevertheless are very compelling mutations, in 97% of these new cases, and 99% overall of ALL cases, through a single whole genome sequencing test, do provide a very compelling mutation to aid the interpretation of the condition. And this is one of the important measures in any approach where we're trying to improve the precision in genomic medicine, whether it's through a whole genome or whole exome or other approaches, is to look at the yield of results, the diagnostic information that is relevant, and in turn, in the case of cancer, the kind of information that may be relevant to treatment in the future. One more paradigm is not directly about whole genome sequencing, but it does indicate that if you have the whole genome sequence characterized from a particular patient, it provides a rich source of information about that patient to personalize the follow-up process. And you can see here, for example, a mutation in the EGFR gene of this particular lung cancer patient. This lung cancer patient responded to treatment with gefitinib, and you can see the reduction in circulating tumor DNA carrying this mutation from 14% down to less than 1% over a period of 2 months. And you can see the corresponding images in the lung of the individual on the top. What you then find in monitoring the patient with the same personalized test that was derived from their whole genome, you actually see, sadly, a relapse, but you can see the measurement of the relapse is quantitative and accurate, such that just approaching 1 year later, the mutation is now present again in the individual's circulating tumor DNA at 1%. And then, sadly, over the following 6 months, elevates to a very high level, 49%. Concomitant both with the regrowth of the original tumor and also additional metastases. So this just illustrates not only the value of having extensive mutation profile, somatic mutation profile of a patient, the ability to use it in follow-up tests which are highly targeted, highly personalized. It also begins to hint at this ecosystem once again, an integrated system whereby you can actually use different methods to apply and understand and better measure the progression of cancer in a patient at different times during their life. I'd like to move on now to briefly discuss some of the technology aspects that we've been working on that are highly relevant. They're growing points in the curve. First of all, clearly technology itself, the actual generation of sequence data, is a very important driver in this whole field. It was a disruptive change in data rate when we first developed the Illumina system based on the Solexa chemistry. And you can see here in recent years, we've continued to use various tools, going to patterned surfaces, faster cameras, improved and faster chemistry, to increase the data rate, which is the overall measure by which we're really trying to characterize these genomes. And you can see a leap there between 2013 and 2014, which was no accident. This was not just a major technology improvement, and this was the launch of the HiSeq X, but it was also stimulated, a development program that was stimulated because of the perceived need in the community and the appetite for moving to larger amounts of sequence and taking whole genome sequencing on up to whole genome levels and to population scale. One of the major impacts, of course, is that as a result of the increased data rate, then the actual cost of the experiment goes down significantly. And you can see here the concomitant reduction in the whole genome sequencing, achieving in 2014 this rather iconic number of $1,000 genome, which is for an essential 30x average depth genome. But it does reflect a very important trigger point for many people's thinking. When previously sequencing was very expensive, among the other limitations, here it is now reduced to something which is very manageable, meaningful in terms of healthcare budgets or per capita budgets. I wanted to return for a moment to the question of whole genomes. I've emphasized the use of whole genomes. That is not to the exclusion of other methods. I've already shown you a targeted sequencing process. But this just summarizes a few points which are worthy of discussion as to why one would focus on whole genomes, or why we focus on whole genomes, and why this also resonated with Genomics England and a number of other collaborators and projects that we are now involved in. It is, of course, the complete accurate genetic makeup of an individual. It is one test, single test, near complete characterization of an individual's genomic information, once and for all in the case of germline. In a cancer, of course, the genome is dynamic and needs resampling, as I've already showed you. Just to explain the second bullet, recapitulating the principle of the Human Genome Project itself. The Human Genome Project in the early stages went through a big debate as to whether to sequence the entire genome or just look at the transcripts. And clearly, if we'd only sequenced the transcripts, we would not have 98% of the information. The same thing is actually true here. In sequencing an individual's whole genome, it really does provide all the information that is likely to be contributing to a condition, even if it's not yet understood. And I showed you the promoter mutations in the TERT gene already. They would come naturally in a whole genome sequencing test. It's also technically simple, a point which is perhaps not necessarily widely grasped as yet. It's a highly reproducible, high-quality product. I've illustrated the trade-offs there, though, of course. There is a question of cost, data volume to be handled, and I'll touch on that in a moment, and the number of samples that can be processed either in a time or in a particular capacity in terms of what is the appropriate way. And the table at the bottom there illustrates just a brief comparison of the kind of information you can get and the level of completeness of the information you can get from a panel, from an exome, or from a genome. In particular there, as well as coding variants, of course, coming from the region that's targeted. But the non-coding variants are restricted to the genome. And the exome, less well than the genome, will also detect rather complex genetic events, consanguinity, uniparental disomy. Some of these events have already proved extremely important in amending a diagnosis for an individual. One of the challenges in moving from basic research to clinical space, of course, is that we are dealing with real-world samples, and just rather schematically they're illustrated here. Blood samples have really been the bread and butter of a source of DNA for many tests, and of course that is now emulated further with the use of cell-free DNA for precise tests. But illustrated also two other ones, in particular the use of FFPE DNA, DNA which comes from formalin-fixed, paraffin-embedded tissue, which is in blocks, which is an extremely suboptimal way of treating DNA, although it's very good for histology. But this has become a major source, potentially, of tumor DNA samples. This DNA is degraded, it is modified, it is damaged. And so we have to look at modifying protocols to see how effectively we can actually generate data, or whether targeted or whole genome, from FFPE material. And I'll just show you a slide on that in 1 second. The other, which I won't show any more on, of course, is biopsies are very important as a currency for collecting samples from individuals. And these, of course, only provide very small amounts of DNA. So it's very important that we continue to learn how to work with smaller and smaller amounts of material, while still getting that all-important, highly representative genome sequence data. This slide just illustrates some current work, collaboration with Genomics England, in working out how to improve whole genome sequencing from FFPE samples. This is a direct comparison between what you see as a whole genome readout from a fresh frozen sample, that's the gold standard, from, in this case, a solid tumor, and you can see a varying read depth and some indication of variance in there. When you look at the same tumor, which has been embedded in formalin-fixed, paraffin-embedded material, you can see here a much lumpier read depth plot and the B allele frequency plots are much less distinct. Really difficult or impossible to make any call about copy number variance from this sample. But by amending the DNA extraction process, and this is one of a number of steps, you can see this same sample, exactly the same sample, can be returned to something which is closely resembling, not quite, but closely resembling the quality of the fresh frozen sample. And this sort of research gives some hope that we can actually learn how better to extract DNA from FFPE samples so that we can get valuable information not just on the single nucleotide variants and the small indels which come through this, but also the copy number variants and structural variants are much more likely to be well read out from such modifications. This just shows an example of developing extraction methods that improve the extraction of the DNA. In a separate part of this collaboration, all the upfront methods for fixing the tissue in the first place and embedding are also undergoing review, and it may be entirely possible to amend or improve the early stages of this process to the benefit of the DNA quality that comes out without compromising the histology information, which is also important. So watch this space. FFPE certainly has been a challenge, but we get good whole genomes from a wide fraction of the whole genome of the FFPE samples. Moving back into more fundamental metrics of the genome for a moment, this illustrates where we keep a very close eye on the quality of the coverage and the bias in every genome. Essentially what you want as far as possible is that the whole genome data you get out is a faithful representation of the DNA that went in, that came from the patient. And each of these blocks of bars here illustrates various segments of the genome that are monitored. Genes and exons are the first two blocks, and then there are some difficult regions, high GC regions, regions which are known to be poorly recovered in certain protocols. And you can see that over the ages, as we go from left to right within each of the blocks, we move from our classic amplified sample prep to a PCR-free prep, and we actually reduce the amount of starting material required. But at the same time, we are increasing the proportionate representation of those difficult regions. So they're coming much closer to the normalized 1, which you see at the top of this plot. 1 means that if you build a 30x genome, you would get, on average, a 30x representation of that particular feature. So you can see, for example, the very high GC-rich ones, they're not up to 1 yet, but they're getting close to 0.7 times what you would expect from a random sampling. And so this is very encouraging in terms of getting really good coverage across most of the genome. But it's an area that we really look at, particularly these difficult areas. Promoters are GC-rich, but we really want to see promoters. And you can see how far we've come. Key drivers are the PCR-free, I've mentioned, improved quality of the chemistry, reducing the low DNA input, and I've already mentioned the use of FFPE. For a more visual picture of this concept, you see here an IGV plot, integrated genome viewer plot of the same genome done in 3 different ways. There are 2 different amplification protocols which work pretty well, the nano versus version 1 and version 2. And this is across a very GC-rich exon, exon 2 of the ARX gene, which is important in medical conditions. But you can see here the adoption of a PCR-free protocol at the bottom does actually allow this very GC-rich exon to be represented in the genome. And so you can see the effect of just looking at these very small points in the genome, which may be medically relevant and are also very important to actually assess how well they are represented in the whole genome, are all part of making a whole genome or other methods really tested and able to provide the information that is really required in the clinic. Accuracy is another important area, and I won't go into this in great detail. There's a whole presentation on accuracy, but I do want to mention this project just in passing, where it makes it important to actually be able to objectively compare different analysis pipelines in adopting ones which are suitable for the project. And this illustrates a project that we developed in order to make that objective assessment. It's called the Platinum Genome Project. It's a public reference data set. We generated deep sequence coverage of whole genomes of each of the individuals in that 3-generation pedigree, and we identified 5 million essentially high-quality variants as a truth set. And we identified them as a truth set because the variants are entirely consistent across every member of the pedigree. So the inheritance test and 1 or 2 other tests essentially passes or fails a variant as being platinum quality. And once we have the platinum variants set, we can then utilize genome sequence data and different pipelines to evaluate how well different pipelines call or do not call the platinum variant set. And the bottom table illustrates a straightforward comparison between the HiSeq analysis software, it's an earlier version in fact, the v3, and GATK. And you can see the recall and precision of either the single base substitution calls and the small indels, 1 to 50 bases, is essentially comparable. You can examine the differences between them. But the other very important point about this is that the reason that the HiSeq analysis software suite was written was in order to cope with the challenge of scale, because you can see here that a whole genome is actually analyzed in 5 hours as opposed to 38 by the GATK suite of software. And this becomes extremely important when we're looking at working at scale, which I'll just touch on a bit later. And these performance criteria were measured on an equivalent hardware platform. One thing that has emerged from the Platinum Genome Project was to actually see clusters of SNPs which appeared to fail the Mendelian pattern. And clearly there's something else at stake here if multiple SNVs in one location of the genome appear to have Mendelian conflicts. And this illustrates one such area. And actually what the reason for this cluster of SNPs having a Mendelian conflict is that in actual fact there's a previously undetected underlying CNV, which essentially, and the read depth reveals this transmitted deletion in the mother. This put us right on to looking in populations more broadly at the same events, because being able to detect a lot more of these complex variants, CNVs, structural variants, and so on, is really important if we're trying to build up a complete picture of the spectrum of variants in a genomes, so that we can then in turn refine the algorithms to improve their detection. And in the bottom part of this slide, I'll just pick out the very bottom track for a moment. This same region in a population of 2,000 Caucasian individuals, these SNPs all show a departure from Hardy-Weinberg equilibrium with an excess of heterozygotes, an excess of homozygotes, which is entirely consistent with this same previously undetected deletion being prevalent in the population. This is moving ahead the slides here. Okay, let me move on. So one other important area which is a focus for medical relevant is the detection of repeat expansions. Repeat expansions may not be so relevant in a whole genome-wide context, but of course in certain conditions, Fragile X-linked mental retardation, ALS, myotonic dystrophy, and Huntington's, these small repeat sequences that expand and move from a pre-symptomatic anticipation to an actual affected individual are very important, and one might at first glance consider that hexamer or triplet repeats cannot be detected by short reads. In fact, they can, and this actually underlies a very important principle. On the left here, you see a whole series of individuals previously typed as positive for expansion in ALS condition, and we detect 95% in the bioanalysis of the short reads. Correspondingly, the normals, some 1,323 unaffected individuals revealed on the whole by our test in the whole genome sequence data, also an unaffected individual, with the exception of 3, which were subsequently retested and actually found to be positive. So the test is extremely sensitive and specific for detecting repeat expansions. This is an important aspect of examining more closely medically important regions of a genome to be able to analyze them in whole genome sequence. What's also very important, however, is that this reflects a fundamental principle where in order to identify repeat sequences, we were not looking at the reads that are aligned to the genome. We were looking at the unaligned reads that are also present in every BAM file, but are not necessarily analyzed or would not be analyzed in the normal process of using an alignment. And therefore, these reads are a rich source of other information about rearrangements in an individual's genome, and they're quite amenable to characterization. And we can write algorithms to extract information such as what you see on this slide. And at the same time, then you can incorporate those algorithms into the pipeline. So we're extracting much more information about CNVs, junction fragments, and these repeat expansions in whole genome sequence data. These algorithms have yet to be folded in most cases, to the pipelines. But this illustrates just one of the growing points in our research, and this is all in collaboration with various clinical and molecular investigators around the world. I said I'd touch on data footprint, and one of the much discussed elements of whole genome sequencing, and indeed any population scale sequencing, is the scale of the information and the amount of data. But this illustrates the important concept of shrinking the data footprint. We start with an assembled genome, a 30x depth genome, which is a BAM file on the left, 120 gig of data. We can do various things, and we have in collaboration with the EBI and other organizations, taken steps to reduce the granularity of the information, in this case reduced quality score bins, to shrink it to 80 gig. And then compression techniques, largely developed in this case at the EBI, the CRAM formula, which provides a 30G version of the original 120G genome. Looking further beyond this, however, we look to the actual concept of extracting the information from the consensus and not storing all the reads. And once you actually take the consensus as read and you examine and harvest all the variants, you form a VCF. These are actually GVCFs, and I don't have time to go into the importance of the G, except to say that these GVCFs store information on every variant for future clinical interpretation, but they also store information on the quality of every homozygous call in the entire genome. And this is very important to be able to call if there is definitely not a variant in a particular position. That's a very important piece of information to know in the clinic, to be able to say you definitely do not have a mutation in this exon, as opposed to saying I can't see a mutation in the exon. So the GVCF is a really important currency, very lightweight, 2 gig of information, sufficient for interpreting, annotating, and interpreting the genome, but it does cover the entire genome, not just the variants. So these last two forms, the 2 gig files, GVCFs with annotation, of course make the genome very portable and easy to transfer. And this arguably is the real currency going forward of genomes. And I've just outlined there perhaps the two preferred forms of data which might become long-term. You might see a long-term storage of the CRAM files to archive and perhaps look up and go back. But really the annotated GVCF, the 2 gig file, you can get many of them on a thumb drive and transport them. They can be uploaded and downloaded. And so this is really the common currency of the genome. And it's important to recognize that shrinking the data footprint has not lost you any information about the variants or about the homozygous reference calls in the entire genome. This really is a portable genome, which really resolves many of the challenges about data storage going forward. I mentioned annotation in passing. Once again, there's much to be said about capturing publicly available and licensed information from research cohorts and from local aggregation. This helps to improve disease association information, and in particular the feature here is that we've done a great deal to work with VEP and related annotation engines, but actually reduced the time again from 12 hours a genome to 3 minutes, and from 10 gig to 0.5 gigabytes of space storage required per genome. So once again, it's important to have very comprehensive annotation, but like the actual analysis before it, it's important to be able to do it in a time scale and with the amount of storage that is commensurate with working at very large scale. I just want to touch on some recently solved cases because we want to get back to the clinic for a moment. And this illustrates how some of the technological advances I've just been describing have had a real impact on diagnosis of rare genetic disease. And these are just anecdotes, but they serve to illustrate the point. These came through our clinical laboratory in San Diego, where we do an undiagnosed disease sequencing service. And we do include all the interpretation and report generation, which is signed off by our board-certified geneticist. Here's one example of an autism-like symptoms in a 1-year-old, where actually the answer was found in a gene that was not previously known to be involved in such a condition, but the genome-wide approach enabled us to actually chase and identify such a mutation, a de novo dominant mutation in this particular case. The second is one where a 9-year-old had had a complex condition from 1 year. And it turned out that a really strong candidate gene for which there was previously nothing had been seen. Because we were now able to see CNVs, copy number variants, we were able to find in this very compelling candidate gene a 55 kilobase copy number variant, which indeed turned out to be a dominant effect in this condition. And the third example is one of a compound het, a recessive condition, But here, once again, the single base mutation had been revealed from an earlier analysis, but actually going back and now being able to look for CNVs, we actually showed that the other chromosome was suffering a deletion, and as a result, the gene was entirely knocked out in this particular condition. This is compelling not only because it illustrates where incorporating the ability to detect CNVs better increases the ability to explain cases, it increases the diagnostic yield, but also in this case, this last case was a 16-year-old, and for 16 years this family had not had an answer. And actually providing a confirmed diagnosis really does a great deal to help counsel the family and really change their lives for the better, even if they can't do much about the actual condition itself. Touching on this interpretation and reporting process as we move towards scale, and this is something of a preview to just talking briefly about working at scale. Sequencing the genome and annotating is now becoming fairly well automated, fairly well scaled, but the question of interpretation and reporting remains a very individual process requiring expert examination. And you can see from this chart, when we started the process in May 2013, that it was taking something like 40 hours per case to interpret some 1,500 genes, looking at all the incidental findings and looking at potential candidates, much of which was to do with the curation and re-examination of literature, and some of which was to do with generating the report, which is the blue pit on the top. And you can see over this period in 2013 and since then, we progressively reduced the time it takes by taking away all the unnecessary steps which individuals were doing manually, to gradually automate, and also very importantly to log findings in a database so that they can be looked up next time round if they come again. And this is the concept of the knowledge base, a very active knowledge base for interpreting genomes. And you can see that today we're down at less than 10 hours a case for a pretty comprehensive interpretation. And now this is our starting point for really continuing to build with collaborations and with hopefully sourcing national experts and international experts. The whole idea of a curation knowledge base could become much more powerful and further reduce that figure so that genomes really can be interpreted, still with the manual inspection and manual intervention, but at the same time in a much more accelerated progress, so that if we're generating genomes at a rate of knots that are really commensurate with the number of cases in the population, we actually do have the ability to interpret and return results through this gradual improvement, automation, curation of knowledge, and the ability to make all this information accessible to the geneticists and other experts appropriately. As an example, and I'm just going to tie in both the first and second part of this talk for a moment, how do you present the information in a way which is really accessible to a clinician? This is an example of an automated report. I've actually chosen a cancer example here, but the rare genetic disease ones are perhaps better known, but they're also more familiar to people. But you can see here we can analyze somatic mutations in cancer in the same way by essentially highlighting genes we know to be important. Here I think ATM and P53 has a mutation in this particular colorectal patient in these genes, a somatic mutation. And there are also some other mutations about which we know less, but nevertheless we highlight them as potentially suspicious, worthy of follow-up or worthy of further annotation. But on the right-hand side of this report, which is a prototype report, we also include tools that actually visualize all the other more complex graphical information I showed you from the very beginning of this presentation. You can actually show in the same report a complete spectrum of copy number variants, gross changes, the context of sequences in case there's something specific about DNA repair defects or something, a clue about the environmental exposures. And the bottom is that rainfall plot to look for contagious events that may be relevant and should not be ignored. So if you have both halves of this report, you really are in possession of a much more complete picture of an individual's cancer genome than if you were simply working with small mutations and trying to correlate them with candidate genes, which is what the left-hand side does. Just moving on for briefly to looking at working at scale now. This is a much shorter part of the presentation. Just to illustrate, first of all, this is very much a thing we've learned with the Genomics England partnership and one or two other collaborations similarly. And I just summarize here the Genomics England 100,000 genomes, the goals of the project, to bring benefit to patients, to create ethical and transparent programs, to enable scientific discovery, number 3, and also to really stimulate the development of the industry. And this illustrates some key features of what we have done in partnership with Genomics England and the NHS, is to develop a program which focuses on cancer and rare genetic disease. It absolutely epitomizes the paradigms I showed you at the beginning of this talk. They are not exactly low-hanging fruit, but they are the group of patients most likely to benefit and where we can learn more about them. We now have an ISO-accredited workflow for this entire process for the whole genome sequencing through to annotated genomes. The interpretation is not yet in that position. And the data are delivered securely and electronically to the Genomics England data center to be utilized for clinical analysis and also aggregation and interpretation. This is the workflow. You see briefly that Genomics England have put together a workflow which starts with the patient, and the patient is seen at a genomic medicine center, and the sample then goes into a central either here at Chesterford in the Illumina facility, or now it's been transferred to the Hingston facility next to Sanger in a purpose-built facility, and thence to the data center in England, which stores the genome and gives access for interpretation. And you can see both the sample flow and the data flow in this process. Just to zoom in on the sequencing process for a moment, because this has been an area of significant concern. How do we handle all the data? How do you convert from a research one-off process to a highly scaled, fully monitored process? This illustrates very briefly the workflow from left to right, from DNA to library formation to sequencing to delivery, and then eventually deletion of the data when it's no longer required at the generation site. But the many steps are really governed by so many steps in QC, and this is the point I really wanted to make here. This is now an industrial process. Most of the time is spent looking at all the steps to look at the passing and failing QC to ensure that every genome really does come through the pipeline and is high quality and is suitable for use. And that's part of the ISO accreditation process to govern this, but it requires a complex industrial process management through laboratory information and also analysis information systems, LIMS and AIMS, which we can discuss more at some point in the future. Finally, just to touch on progress, there's some 13,000 samples have been delivered. The majority are rare genetic disease, not least because of the additional challenges of working with real-world cancer samples. And this project includes both Genomics England and an earlier pilot at the University of Cambridge, or UCAM, which was one where we were really fortunate to learn many of the lessons with a more local collaboration. And the building at the bottom illustrates one other important point, not just a purpose-built facility, but this was a Wellcome Trust contribution, so this is other sources of support coming to help the project along with the building of the Ogilvy building at the Hingston campus. Finally, to draw conclusions, first I'll show you once again the slide of the clinical ecosystem, just to remind us that while I've taken you in some detail in some areas to the details of what the individual bases are reading out and how we align the reads and check for accuracy in every genome, this is all part of the process. This is all within one of those colored arrows. This is generating one piece of data, one piece of patient information, which is then stored, either the entire genome or, as I mentioned, the portable GVCF is perhaps the more portable version of an individual's genome, which gets stored in a knowledge base and then gets aggregated. And I showed you examples where researchers are using aggregated genomes to do essentially straightforward things like identifying CNVs in the population that we couldn't see before. And these are very straightforward and valuable uses of aggregate DNA information. And of course, the much more big challenge is to aggregate the clinical information alongside that and be able to perform really good genome and phenome interpretation analyses on aggregate genomes to really help our clinical interpretation of variation in the genome. Finally, to sort of summarize the next steps, if you will, just some of them. Of course, there are many next steps in this whole process. It's important to reiterate that, among others, the Genomics England project is there to demonstrate the utility of whole genome sequencing and perhaps to evaluate the utility of whole genome sequencing for cancer and for rare genetic disease. Clearly, we're still looking at the relative value of this as part of an integrated system and how it compares to other approaches to gaining genome sequence information from individuals. But suffice to say, some of the benefits of the whole genome sequence, despite the size of the data, which I hope I've talked to you a little bit about, and the technical challenges, actually it's a very simple workflow and it really is one test fits all. And I think are confident that the whole genome sequencing approach really is the way to a very high diagnostic yield, Once you have all the information, you can interpret it. If you only go for some of the information, you don't know if the answer is in that information or not. And that's why I put forward a simple philosophy for whole genome sequencing. I've mentioned aggregation of data. We need to learn how to do that much better and at scale. And we also need then to look at the impact on healthcare, to build economic models and initiate the reimbursement from healthcare systems. We have some interesting collaborations going on. In England, of course, it's a state-funded health system. In the US, it is not. And so reimbursement is a very different question and requires different evidence data sets to actually help decide about the value and utility for reimbursement. And we have some interesting collaborations going on in that regard, particularly with rare genetic disease at the moment, but possibly soon with cancer as well. I'll just mention one of the other interesting challenges, of course, is that some people have said that, well, as you can diagnose so much more, it's going to cost so much more to follow up all this information and treat the patient. That's a very important element to take into account when looking at the economic models and how they impact healthcare. The converse, of course, is the advantage. You're much more likely to get a precise and hopefully correct diagnosis in more cases, which ought to make a difference in a positive way. I've mentioned ecosystems. I've not mentioned other sources of data, which are very important. I've touched on whole genome and panels and circulating tumor DNA. I haven't mentioned RNA data, which we, of course, do reckon is very important in cancer analysis, and the ability to look at information shared across networks, really to enable national adoption and international adoption, requires this continued concerted effort to build this very model that I illustrate at the bottom of this slide. I'll just finish with a list of names for acknowledgements. I'd particularly like to acknowledge both all the colleagues in Illumina, and there are many more who are not listed there, but also very important collaborators. My group and much of Illumina works almost entirely through these collaborations, and very key collaborators are listed there in various ways who have contributed to individual disease studies, or the scaling process, or the accuracy of genomes and variant calling, how to look at cancer, and so on and so forth. So I'd like to extend my thank you to those acknowledgements, and also thank you for listening to this presentation.
Thank you so much, David, for such an informative talk. We do have a number of questions, but before we start on those, I want to remind our audience how you can submit your questions. If you can go ahead and submit them by typing them into the Q&A box, which you can find on the green Q&A button on the left-hand side of the presentation screen. Okay, so first question. DNA mutations were well covered. However, I'm curious if you think that histone modifications or DNA methylation could be relevant to cancer diagnostics.
Good question. I'm sure it can be. I think one of the reasons that I emphasize the study of DNA mutations is quite simply we know more about them. It's a very extensively studied field, and of course with all these areas we're building on prior research. Our ability to understand the mutations really predicates what we're going to search for first. I think we understand much less about methylation at the moment. There are some interesting and compelling insights emerging from methylation patterns. And we have some collaborations going on a smaller scale to look at methylations in whole genomes of cancer patients of our own or of our collaborators to see if we can gain some insights or help them to gain insights. It's worth reflecting in the case of methylation, in terms of DNA methylation, you mentioned histone methylation, but I'm going more on the DNA methylation at the moment, is that actually, of course, there are forms of methylated bases which are really previously undetected and only been discovered a few years ago. So the work of Shankar Balasubramanian, for example, the Solexa founder, has actually uncovered the importance of hydroxymethylation. So we are faced with looking at methylation and hydroxymethylation and other rare modifications of DNA. So it's very much a research area, I think, at the moment in terms of what are we looking at? Are we even looking at the right thing? And once we have that, clearly the power to sequence large amounts of DNA and methylated DNA can be more rapidly transferred into beginning to look at the clinical information. There are others, I'm sure, in the audience who know much more about the clinical information of methylation than I do, but I certainly look forward to exploring that area much more.
Okay, so there is another question related to epigenetics and epigenomics. When will the power of genetics and genomics be turned to that? I guess if it's still in the research phase, it's still a little while away.
I think that's right, and I think because it's in research, different people will take different views as to when or how to address the question. Okay. There are some small regions of the genome. People have looked at individual positions for methylation status, And if there's a definite test which really correlates well in a single part of the genome, there's every reason to go ahead and start to look at that in a more clinical situation. I expect there are already examples of that I'm not aware of. But it is clearly going to be different for different projects.
Okay. So another question, when focusing on somatic mutation studies, is it appropriate to process normal versus infected tumor tissue sample or only tumor tissues?
So tumor normal versus tumor
Yes.
only, is that
Yeah.
there was an infected in there somewhere, which I think maybe...
Appropriate to process tumor normal versus infected tumor tissue.
Okay. Still not sure where the infected comes in, but let me answer the tumor normal versus tumor question. I think, yes, clearly we derive a great deal of benefit from looking at the normal as well as the tumor in every patient. We do find the sensitivity to eliminate germline mutations from our putative somatic mutation panel requires that exact match for optimal subtraction. We don't want to be looking at somatic mutation collections where a significant number of the mutations may still be germline. We actually want to know that we are looking at mutations that are absent from the normal DNA of that same individual. And so the tumor normal pairing is very important. Yeah.
Okay, sorry. So this next one we can probably talk about for quite a while. What are the current major challenges to use genome sequencing as a common diagnostic tool for large patient population?
It's the Genomics England question. And in particular, I should emphasize, the Genomics England project is a pilot. It is not a population scale yet. I think we reckon that perhaps some 5 to 10-fold the scale of this project would be required if we were to address the same groups of patients at a truly population level. I think clearly, on the positive side, we've already made leaps in terms of the cost and efficiency, and I'd like to say the quality of the sequence information as well. It certainly does provide many good diagnoses. We continue to work on the areas that could be improved, as I've already mentioned in the talk. So cost data generation perhaps becoming a very viable proposition, and the cost of it falling well within reasonable budgets on a per capita basis compared to the amount of money that's being spent on other aspects of a particular disease, for example. So cost can still reduce, of course, and that's an important area that as demand increases, one wants to make this kind of assay all the more available and not limited by cost alone. But quality is extremely important, and we maintain that. The other areas, of course, are outside the central core, the sequencing engine. And these are areas where we work very much with our collaborators to examine it. The ability I've already mentioned to collect sufficient good quality DNA to start the process off. So we've had to completely re-examine how the formalin fixation process is done. It's been done that way for 100 years or more, but we need to change it now because there's a new demand for it. Or do we move away from FFP? These are big, fundamental, essentially roadblocks to the process because you do want every cancer patient to benefit. At least you want every cancer patient not to be excluded simply because their sample wasn't good enough. You at least want it to be a choice that it is not the best test to apply to that patient instead. Downstream as well, I think I've mentioned the ongoing process to resolve the data management challenges, shrinking to a portable genome, scaling up cloud-based infrastructure, deciding whether this is going to be in a public cloud or private, which substantially changes where you have to invest your infrastructure effort. And of course, the entire process of interpretation. That's a long one. I think what I've hopefully showed to you is the early signs, the glimmers of diagnostic yield being significant for a significant number of patients. And any one patient, some of them have been waiting 16 years for an answer. Cancers have actually been turned around as a result of a cryptic rearrangement that couldn't be detected, that was eventually detected by whole genome and dictated a change in treatment, resulting in significant remission. So these anecdotes are real, they can be accessed, and we do want to really remove these roadblocks so they really can become accessible to more and more patients, when in turn we will then learn more and more to continue to increase the diagnostic yield. In the case of cancer, of course, there's another whole area of difficulty. With rare genetic disease, a diagnosis is a very suitable endpoint, a very good endpoint. With cancer, of course, well, you already know you've got cancer. It's how you treat it. and what the prognosis is. And this is a more complex interpretation problem, which is going to take longer to resolve. But one way to do it is to stimulate it with high-quality, systematically collected data in the first place, in that all-important knowledge base, so that we can really begin to look at these other hurdles that need to be overcome if whole genome sequencing, or any large-scale sequencing, is to gain maximal use in a population setting.
So you talked quite a bit about cancer there and gave a lot of examples in your talk. So there's a question that says, given that genetics and genomics tend to be currently focused on cancers, could you comment on which diseases will be the next areas of intense study?
Great question. And that list left out the rare genetic disease, which I hope is, of course, a given as well. But it's very important. I think if you look formally at the involvement of some genetic factor, be it a risk factor or modifier or a pharmacogenetic variant, there is some genetics in almost every condition. Some of them may never achieve real significance. They're very multifactorial diseases. Perhaps the better way to manage it is to work more directly with lifestyle and measurement and modification. But the fact remains that genetics and genetic factors are influential in many diseases. And as we collect more data through studies like this, we will actually improve the predictive value of genetics for some of these conditions. If I was to pick out a particular area which is of interest, I think anyone who's looking at a particular disease type, I'll pick out the cardiac conditions for a moment, because the cardiac conditions actually embrace a spectrum of some Mendelian conditions through to some very multifactorial conditions with strong environmental factors. But therefore, if we start to look at the cardiac conditions and sift out the Mendelian ones and find the genes which are important, which may also have minor effects in the more complex conditions, we can begin to gradually use the accessibility of the entire genome to actually dissect out the different genetic components and begin to measure what the contributions are, as well as finding ones which are previously not seen because we were using a more targeted approach previously. So I think there are conditions like that. I think many conditions do have Mendelian phenocopies, which may or may not give insights to the cause of the complex conditions. But there is a rich area for dissection of the genetic components through whole genome, which will evolve over a great deal of time. But I do think these data sets promise to give investigators or experts in these common and complex diseases a great deal more to work with in terms of finding candidates that can then respond to, indeed perhaps require the development of more refined statistical tools to analyze the data in the first place.
Okay. So staying around the rare disease, there's a question asking, what are the challenges in making a diagnosis for a rare genetic disease?
What are the challenges? I should first say, I'm not a clinical geneticist, so I've not done it myself in practice, but I can certainly reflect on a few of the challenges. I think clearly the ability to ask the right questions in an undiagnosed disease we're talking about here, I guess, for a moment, where we don't know, we might be looking at a mutation in an existing gene, but we don't know which gene it is. Or we might be looking potentially at a mutation in a gene that has never previously been involved in a disease. These are two different, but also both quite difficult problems. I think it's understanding how to address the challenge. It is important to look at all the candidates that may previously have been involved in disease. It's also important to look at generic questioning of the information. Am I dealing with an autosomal recessive? Am I dealing with a de novo? Have I examined the parents in the case of demonstrating a de novo? And looking at the different ways in which one can address the question. Not an easy approach to describe, or even for me to describe, especially. Others in the audience will be better qualified than I am. But I think that the bottom line must be that the process by which these people are addressing these problems is successful, because undiagnosed diseases involving novel genes and mutations that are not straightforward to find in the first place are actually being interpreted and forming a very convincing diagnosis and or stimulating functional follow-up. I think one of the interesting things is also that if a new gene is discovered, a colleague of mine who works with me, Ryan Taft, in very early studies of a leukodystrophy family, discovered a gene that was not previously implicated in disease. But that then led to a search for, is this one right or wrong? Is it right or not? And within months, they then had several more cases of the same phenotype with mutations in the same gene. And that's how this process moves from being very difficult and very uncertain for the first case. You think you've got it, but have you really? Then to build up the either functional or in this case population-based confirmation, the first finding, albeit uncertain, can really open up a new area of investigation that can very rapidly lead to confirming the discovery and actually making it then much easier for future cases of the same phenotype, patients like me, which is becoming something of a phrase, to be more easily interpreted. That's how the knowledge base grows in a very simple way from the rare genetic disease examples.
Okay. So on to a slightly different track now. Can you comment on the utility of using GWAS in combination of whole exome sequencing as a lower cost option to whole genome sequencing? So I guess they're talking about combination of GWAS studies and exome studies versus whole genome as a lower cost option.
I'm not sure I understand really where the question's trying to go there. I could answer that in several ways. So
okay. How's about
perhaps if the questioner would like to make direct contact, we could have more of a dialogue on that one.
Okay. It appears that the genome mutation signatures indicating exposure has potential application in public and global health settings. Are you aware of any research or work in this area? I guess there are loads of sort of not population but large-scale genome studies that are happening off the back of Genomics England.
Well, Genomics England is still at a very early stage yet. I think we're talking about cancer exposure, the signatures of exposure in cancers, the smoking and the UV and so on.
Yeah, they just say genome mutation signatures.
Genome mutation signatures. Let's assume it's that, and I would really again refer them to our collaborators or people who've gone beyond our collaborations. So particularly Mike Stratton has been one of the people I know well who is really investigating the importance of signatures. I think much of it is still in research and observational. I think the clinical significance, I think will take some time, but I feel compelled that information should be part of a report so that in the future, if people are looking at aggregates of cases, they can take that into account in the interpretation.
So I guess when it comes to looking at that data, sort of a related question is, will the sequencing detected in the UK, I guess as part of this GEL project, will be available to other parts of the world, such as the US, for further research?
That's a question for Genomics England. And I think it's a much wider question as well, because I think I would almost say everybody, but perhaps most people anyway, would see the value of sharing information of both genome, genetic, DNA sequence information, RNA sequence information, and also clinical phenotypes, in a way that for the moment has to address the question of privacy of the individual, so the data has to be shared anonymously or in an anonymized way. So I think the opportunity is there to do it, and I think one of the great opportunities that we have, starting within Genomics England, certainly the model is there to share it nationally and to make it accessible while ensuring the protection of the data and of the individual. That's the basis on which the consent is given for each sample to be donated. But undoubtedly the mechanism for sharing within a particular national organization is there, and indeed the standardized nature of genome data output provides an excellent platform for cross-comparison. If only phenotypes were equally standardized. And I think there's now a significant move towards tackling the much tougher problem of systematizing phenotypic observations in a way that the same sorts of patterns can be drawn using phenotype data as well as genome data from these cohorts. But you mentioned international as well, and that's a very interesting question. Because I think, particularly I think some time ago we polled pediatric genetic departments, would you be willing to share your data with other hospitals? And of course they say yes, because they may see one case in their lifetime of a certain type, and then they have to make a judgment on that one case. But if they were to network with 100 other hospitals, they would suddenly have 10 or 15 cases the same. And this networking process is already happening, and there are apps which are encouraging that communication. But clearly, to take that concept further, there is great value in aggregating from multiple sources because the diseases and the causes of disease are essentially probably mostly the same worldwide. So it's absolutely valid to pool the information in a way which people find acceptable, either with national boundaries or consent and privacy issues, and it should be done. And certainly the very digital nature of DNA information allows for DNA itself to be a great currency for real comparisons between genomes. and perhaps that can in some way help the formalization and the standardization of this data sharing. I think if we can respect national boundaries or particular ethical policies or government policies in different countries, but nevertheless build the mechanisms by which those boundaries can be jumped over through permissions, through granting permission, through asking permission, whatever it may be. We might not see a single database for everybody in the world with a particular disease, but we might see a term which is often used as a federation of data and sharing across boundaries. And I think the very fact that we have mechanisms for data sharing, cloud-based computing, very fast communication, of course, takes away the technical challenge of this. It can easily be shared technically. The question is how to structure it in such a way that everybody finds it acceptable and it doesn't become restrictive to the final potential gains.
Okay, so we have time for one last question. So sticking with the ethical and the data sharing there, how will this project exactly create an ethical and transparent program? You touched a bit on the ethics there. What about the transparency as well?
The transparency, which is a really important part of the Genomics England mission, and again, something that they've really taken the lead on for this project. I think quite simply, or to put it simply, it's not simple, but to put it simply, is quite simply ensuring very good engagement of the public, the general public and the professional, medical professionals and so on, so that they are aware of what is being done and why and how. And I think it's very important to reflect on the fact that it is not even just what you actually do, it's the perception of what's being done that is important to be straight. And if you're completely transparent, the perception matches the reality. But if there is not transparency, there is fear. There is fear of the unknown. What's happening to my data? And I think that's a very important part of being transparent, and that is the public engagement process, explaining what's happening, being very obvious about protecting the data. The consent forms and the consenting process does require informed consent. I've not been part of the consenting process, but the ability to make it clear to the individual so they know what they are consenting to and what's going to happen to that data is part of that transparency. Just as being very clear about how they store the data. And various phrases, I think, stick. Genomics England have chosen to store the data in a private cloud system. It is not on the public cloud, so it is free from some of the risks that are associated with public clouds. And that creates both a perception and a reality that there is a level of protection and security and responsibility has been taken for the data. Another example is the data is viewed as it's a reading library, not a lending library. You can gain access to the data and you can read it. You cannot take it away. Right. And so that's perhaps a simple way of being transparent about how you handle data by using a very common analogy, the reading library, where you can't take anything out, versus the lending library, which is a philosophy employed by the Genomics England data center. Clearly these things may change, but if we change to become more accessible, it has to be through essentially the public being as much involved in that decision and seeing the benefits versus the risks. And I do see, and people have predicted, major changes in social attitudes to privacy and data. People have already cited the change in privacy since Facebook and other social media. People are not actually that worried about privacy, or they get hurt and things move on. And clearly we may see changes, but they must be changes which involve the public understanding of what's being done here, and their acceptance, indeed part of the decision-making process, to actually see data made accessible for public good and for medical use. Well,
thank you very much once again, and we did have a number of questions that we didn't get to, so I really apologise for that. Do you have any final comments or takeaway messages before we wrap up?
No, certainly I hope that many of you are stimulated to see both the challenges and the appetite to actually help resolve and really implement this very exciting piece of technology into healthcare. I do not for a moment suggest that genomes are the answer to everything. But they are a very important element of every one of us, which contributes to our life and potentially can be utilized effectively in healthcare. And I hope that benefit can be realized really in a population-wide scale. It takes all of us, and it takes collaboration, and it takes time. And I hope you're very much involved in this. I'd like to thank you all again very much for your time.
And today's webcast will be available for on-demand viewing for the next 6 months. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement on to your colleagues who may have missed today's webinar event. Thank you very much for attending. Thank you once again, David, and enjoy the rest of the event.